So this talk is going to be a little bit of a mix of some very basic principles. One of the I think, uses of mouse genetic analysis is to illustrate some things that we don't necessarily get in human genetics these days because the technology has sort of outpaced the fundamental methods of genetics that have historically been applied. So you'll see that. You'll see a little bit of just the history of science, and you'll see some of the applications to present-day genetic challenges, and also in this version of the talk I've included a little bit on genomic modifications, which is a practical application of mouse genetics, since that's particularly relevant to some of the discoveries that we're making in human genetics these days, and you may be interested in that. So, you know, genetics is a well-established, although not that old, a discipline, and we're going from Mendel's discovery of segregation and inheritance in peas to the kinds of highly technical genetic analyses that we do in the characterization of mammalian genomes and other genomes. Genetics as a discipline, there are two strategies for understanding gene function. One is to start with the phenotype and then try to figure out the underlying genes that contribute to that. That's very much the nature of the kinds of studies that are done in human genetics, where particularly we're interested in disease phenotypes. And it's also historically been the basis of what's done in model system genetics, usually looking at what are monogenic mutations, which can inform various aspects of biology, in particular, for my interests, developmental biology and organ development. The alternative strategy is to do what's variably called reverse genetics, in which one identifies a gene and modifies that in some way and then studies the consequences of that. And I'll be illustrating that at the end of the talk. So, as I mentioned, you know, Mendel discovered genetics for us, basically, and one important aspect of his discovery was the law of independent assortment, and whether that's because he was amazingly fortuitous with respect to the traits that he chose, or whether he ignored the anomalous data. The reason that he came to the conclusion that genes assorted independently is because he happened to choose traits that were all on separate chromosomes. And so they did assort independently. But we know that independent assortment is not necessarily the case for many traits, because traits do lie on the same chromosome. And when you get independent assortment, as shown here for genes that are on the same chromosome, we know that genes on, for genes on different chromosomes, we know that genes on the same chromosome, by definition, do not sort independently because they are in the same unit of inheritance. And so when you see the non-random distribution of traits in the progeny of a cross, you can begin to make conclusions about where the genes that underlie those traits are relative to each other. And again, this requires that things show some very strong evidence of monogenic inheritance. Otherwise, the analysis of this really becomes too confusing. But when you do have genes with strong effects, you can follow their inheritance, and you can draw the inferences about their relation to each other on chromosomes. And that's illustrated here, where you see 3 different loci, and what is important to appreciate is that because the chromosome is a physical unit, because recombination occurs for our purposes, randomly across the chromosome, the position of genes with respect to each other is a function of where crossovers occur, or rather our inference about the position of genes with respect to each other is a function of where they occur, and therefore a function of how often they separate in a cross. And so where you see genes that are close to each other, the recombination events that separate them are going to be rare, and where genes are farther apart, the recombination events that separate them are going to be more common. And our measure of genetic distance, centimorgans, is simply a function of the frequency of these recombination events. So what you need to keep in mind that a centimorgan is a biological unit. It's a measure of a frequency of basically of a crossover. Now, fortunately for genetic analysis, as I said, recombination at a macro scale is random, and so centimorgans are proportional to the physical distance on the chromosome, and certainly they're proportional to the order of genes on a chromosome. And so we can look at the distribution of traits in the progeny of a cross, and we can draw conclusions about the position of genes with respect to each other. And this is the principle of linkage analysis. So we know that after Mendel and the rediscovery of Mendel's laws, genetics began to take off in model systems, particularly in Drosophila by Thomas Hunt Morgan. What you may not appreciate about mouse genetics is that the history of genetic analysis in this system goes back pretty far as well. And this is actually the first genetic analysis, or an illustration of the first genetic analysis in the mouse, where the pink eye mutation and the albino mutation were crossed. And what you would expect is that if these are non-complementing mutations, that is that if they're different loci, these were both known to be recessive, what you're going to end up with a heterozygous. And so the phenotype of the mouse is basically the wild type. But presumptively then, any of these mouses is then heterozygous and carrying the mutation. And so then when you do you cross these two heterozygous parents together, what you expect is that a roughly 1 to 2 to 1 distribution if they were independently assorting, and that you see here, in which you see the heterozygous parent and the two parental types, but a small amount of mice which actually showed both of these phenotypes. And the only way that this could occur is as if a recombination event occurred between these two loci, and you ended up having them both on the same chromosome, and then getting two of these recombined chromosomes in a single individual. And as I said, you can use a calculation of the frequency of this to determine what the position of these two loci are with respect to each other in centimorgans. And just to make the point, this experiment was done in 1915 by some people whose names you may recognize. So there's a long history of genetic analysis in the mouse. And as loci were identified and as these crosses were done, what one built up is a linkage analysis, or a linkage map of the mouse. And again, what you have to appreciate, this is a purely biological entity. There was no knowledge, in fact, of chromosomes at the time. What we had is these things were called linkage groups. And genes were put into linkage groups by making crosses and looking at their inheritance with respect to other mutations. Very tedious process, particularly because the phenotypes of the loci were quite diverse. They were morphological phenotypes and coat color phenotypes, like the ones that I showed you. You'll see up here, there's the H loci. Those are histocompatibility phenotypes found by looking at aggregation of blood cells. There are isozyme phenotypes identified by running enzymes on gels. And you have to appreciate each of these represented a different assay. An extraordinary amount of work to just sort of put things onto linkage groups. So there are two things that really revolutionized the field of genetics. The first is the recognition in, I believe, 1954 about that chromosomes were the unit of DNA inheritance, and that these linkage groups corresponded to chromosomes. So now we have a reason for why things were inheriting non-randomly. This is because they were on the same physical entity. And the other recognition was more recently that one could get away from looking at each of these different assays by assaying the underlying variation that caused those variable phenotypes, which of course is the variation in DNA sequence. So all of those isozymes ran differently because they had different amino acid sequences, and they had different amino acid sequences because they had different DNA sequences. And so Botstein and his collaborators recognized this and put out there that let's skip the isozyme analysis, let's skip the histocompatibility analysis and look directly at the DNA. And beyond the theoretical insight that this embodies is the technical insight, is that by looking at a single assay, the measurement of DNA variation, we could now have one way to basically analyze loci all over the genome in a relatively efficient manner. And that assay at the time was using restriction enzyme polymorphisms, and then these would then have to empirically be used to find variation. You had different enzymes, and that worked reasonably well, but was not the most efficient way to develop, to identify polymorphisms that could be used for linkage analysis. But then with the development of microsatellites, which were PCR assays that one could do to look for variation across the genome, and then SNP assays, we have very, very powerful, rapid, and importantly automatable ways of assaying DNA sequence variation across the genome, and that allows us to do linkage analysis in an efficient manner. A little bit of local history with respect to mouse genetics relates to the development of inbred strains. And this is one of the important things that makes mouse genetics a very powerful tool for genetic analysis, is that unlike many model systems and unlike the human population, obviously, we have mice that are genetically homogeneous. And the origin of that was work done in part by Castle. Castle was actually a plant geneticist. He worked at the Busey Institute, which was an institute affiliated with Harvard. You may wonder where this is. This is actually at the Arboretum. This beautiful Gothic building is no longer there. If you've ever been to the Arboretum, you've seen it's been replaced by this Soviet-style bunker. So this is not exactly progress. But this is where the plant geneticists affiliated with Harvard worked. Castle made a brief but very important foray into mouse genetics when he and others recognized that if we had a model system which was genetically homogeneous, we could do some interesting things. There weren't the issues of tumor rejection, and he began working with a woman, Abby Lathrop, in Granby, Mass. Granby is in western Massachusetts near Deerfield. Abby Lathrop was a retired school teacher, and she was raising mice basically for the pet trade. So this was back in the day, people would get all these different mutant mice that had interesting coat colors and interesting fur and have them as pets. And she was supplying them. And Castle and Snell and other investigators got a hold of her, asked her to do inbreeding. And these are her notes, actually, as she began to do crosses to inbreed mice. And as you'd expect, of course, is that as you fix recessive mutations, you're going to lose viability, you're going to lose fertility, but enough lines survived, tolerated it, that we now have very robust, long-lived, inbred lines of mice which are genetically homogeneous. And so this is a very powerful tool that allows us to do genetic analysis. And I'm going to give you an example of some stuff that we work in my lab. We have a model of polycystic kidney disease. This is a recessive model. The human disease is a dominant in most cases. And you can see the mouse mutant here. We call it JCK for juvenile cystic kidneys. And what we did to identify the mutation was linkage analysis, taking advantage of mice that had inbred backgrounds. So when we're doing association studies or linkage analysis in non-inbred populations, they're doable as long as you have informative polymorphisms. You can do your linkage analysis, but you have to look at the parents in order to find out what the informative loci are. When we're doing this on inbred mice, we know what the loci are. They've been characterized, and so we know without testing the parents that we can look for and find informative markers, and we can do crosses. And so that's illustrated here, where we took the JCK mutation, which was on the most commonly used genetic background, C57 black 6, and then this is crossed with a different strain. You then end up with the F1 generation, and then intercross those. And with the F2 generation, what we're looking for is mice that are affected. And then what we're doing is testing markers distributed across the genome, and we're looking for those that are non-randomly inherited with respect to this mutation. And as I said, if you have random inheritance, that means that those markers are on a different chromosome. If you have non-random inheritance, it implies that they're on the same linkage. Well, it defines them as being on the same linkage group and implies that they're on the same chromosome. And then you can measure the frequency of those recombination events or the non-randomness, and that then allows you to draw conclusions about position. And so this is what we did many years ago, looking at microsatellite markers, and you can see that we have parental alleles, and they're this particular marker is segregating randomly with respect to the mutation. All of these are affected mice, and in this case we have a marker which is segregating non-randomly. It's not at the mutation because you see a couple of heterozygous mice, so these are affected mice, but they're heterozygous, so it's close to the mutation, but not at the mutation. And then if you test multiple genetic markers across the chromosome, and you see the MIT refers to work done here, basically, to identify genetic markers, and you calculate the recombination events, and you can then put things in an order, and then you can define a genetic interval in which the mutation must reside. And this is the recombinant interval, and then you can go into that interval and you can look at the genes, and at one point you had to go and fish them out themselves, and now you just look on your genome browser and you see what's there, and then you do your sequencing analysis and you find your mutation. So this is linkage analysis in model systems. It works great. And you can do all kinds of monogenic analysis using this approach. What we've done more recently is take advantage of SNP mapping, develop panels that allow you to localize mutations relatively easily. You see the different colors represent the different parental strains. The yellow represents the heterozygotes. And in this case, we're looking at a mutation that was on the black 6 background, which is represented in red. These are affected. So this is effectively the same analysis that I just showed you with microsatellites, but you're looking at a genome-wide analysis in one shot. And you can look for your region of homozygosity, and then you can sort of look more finely, and within that region of homozygosity, you can see the same recombination map that I showed you that was inferred. Here you're actually seeing the analysis directly and localizing with a relatively small number of mice a mutation to a small genetic interval, and then this genetic interval corresponds to a physical interval, and once again you look for your genes in there and you do your positional analysis. So this has enabled forward genetics in a very powerful way. One of the tools to do forward genetics in the mouse is to take advantage of ENU mutagenesis. ENU mutagenesis was really developed as a method in mouse genetics by Bill Russell, who was working at the Oak Ridge National Laboratory. Oak Ridge always had a big biological research section studying the consequences. So Oak Ridge was the first peacetime atomic energy laboratory. The biological section studied the consequences of radiation. They were looking at a lot on effects of mutation by X-rays, and Russell extended this into the effects of mutation by chemicals, and established that a chemical called ethyl nitroso-urea, when given in a particular protocol, was a very effective mutagen in mouse, could be tolerated by mice, would generate a mutation frequency of 100 times the background frequency, and allowed you to basically create mutations rather than waiting for those spontaneous mutations such as pink eye and albino that I showed you. And so this has led to forward genetic approaches in the mouse such as this, which is what we do, in which you treat a mouse with the ethyl nitroso-urea, and then you cross it to a wild type. Your first generation progeny is by definition heterozygous for any mutations that were generated in this G0 parent. If you're interested in heterozygous phenotypes, you can study these. If you're interested in recessive phenotypes, such as we are, you need to do some additional breeding. So you take your G1, cross to a wild type female, you take your generation 2 daughters, back cross to dad, generally a bad idea, and in the third generation you have your affected progeny, and you look for what other phenotype that you're interested in. So this is a classic phenotype-driven analysis, and then we use in our studies, we're looking for late embryonic phenotypes as models of congenital disorders. We actually do the cross as an outcross, so we're introducing genetic informative into the cross, and then as we obtain these affected individuals from multiple related parents, they can go right into a genotyping queue, and we can do positional cloning of the affected mice, and it works very well. So we're able to get lots of interesting phenotypes, and the genetic analysis goes very quickly, particularly these days. Since we have high throughput sequencing techniques, we don't need very fine resolution to begin to look for the genes that are affected. So you combine genetic mapping with exome sequencing, and this really works great. Once you have a point mutation, you need to be sure that what you see is causing the phenotype, and just because you found something doesn't mean that's the causal mutation. And so there are a variety of tests that you can use to establish that the mutation that you found in fact causes the phenotype that you are interested in. So that's monogenic mutation. That works great. But we're interested in not just those, but complex traits. There are a number of reasons why complex traits are complex. And studying them in a model systems, in theory, has some advantages, but as it turns out, has turned out to be quite challenging. So I just want to show you that. So I showed you the JCK mutant phenotype, and I wasn't entirely forthcoming, because in fact what the phenotype looked like when we began to study those F2 progeny was more like this. These are affected individuals from a single litter, and they are obviously showing polycystic kidneys, but they're obviously showing a very different range of severity. And the reason that that severity is different is because presumably of modifying genes which affect the progression of the disease in these F2 mice. And the F2 mice are segregating the mutation, but they're also segregating these modifying genes. And what you're seeing is the consequences of mice having some of these modifying genes and not others. This is Mark Daly's method of doing quantitative trait linkage analysis many years ago. I think he was in junior high school when he first established this. And these methods were used to basically connect a continuous trait, a quantitative trait, with genetic analysis, and basically then make inferences about probability of where the loci affecting that quantitative trait resided on the genome. And so this is all now a probability analysis, and it allows you to then basically calculate a LOD score for loci with respect to the trait that you're evaluating, and that is shown here with respect to the modifying trait that affected the progression of disease in the JCK mouse. And so this was great. It actually illustrated some of the presumptive power of mouse genetics for doing QTL analysis. And that power was that if you had defined genetic backgrounds, you would be able to reduce the complexity of the loci that contributed to phenotypes. And if you could do that, you could do genetic studies and positionally cloned genes. And so this is consistent with that model. If you have humans who have a variation in polycystic kidney disease, you're not going to get this type of LOD score for a QTL. And so that was the driving theme in QTL analysis in the mouse in the long term. But the problem is that this is just a QTL peak. And as you can see, it's fairly broad. This is the length of the chromosome. It covers a lot of it. And so finding the underlying genes remains the challenge. Now, again, mouse genetics, in theory, has some advantages because you can do crosses. You can take mice that have particular regions and you can ask questions about them, and that's called a congenic analysis, where you're taking specific bits of the genome and putting them on a different genetic background and asking what's the consequences of that. And so here's a region in which the QTL was originally localized, broken up, then it's localized into this, and then you can do sort of serial crosses to try to narrow this. And so in theory, this works great. In practical terms, this is extraordinarily painful. And trying to find these different bits and test them each individual is truly inefficient. And even at the end of the day, when you do this and you can localize these bits to a few megabases, they still have many, many genes. And so the challenge has been to, within these regions of strain discordance, finding the actual genes that cause modification phenotypes. Sometimes we're interested in traits that are not necessarily a modifier of a mutation, but are just a strain-specific traits. And mice strains vary in all kinds of different aspects. This is an illustration of an airway hyperresponsive phenotype. This is a model of human asthma. It's basically how twitchy your airway is in response to a sympathomimetic challenge. And you can see that there are some strains which are quite twitchy, and then some strains which are not so responsive to these treatments. And then you can do a cross, and you can see how this trait segregates. And there are methods of measuring the airway hyperresponsiveness, and this just illustrates that in our hands, there is clear segregation in the strains, and then there's an F1 shows a hyperresponsive phenotype, and then by making a cross, you then can look in a back cross generation, the N2, and look for segregation. And this was my first collaboration with Lander, and using the tools that Mark developed, we were actually able to localize QTLs corresponding to this airway hyperresponsiveness trait in the mouse genome. And again, this is not likely to be a Mendelian trait, and you can see that multiple loci were segregating with this phenotype. But you can also appreciate that these loci are really, really large. And so there are thousands of genes underlying these peaks. And it's fine to map loci, but what we're really interested in is genes. And if you can't get to genes from loci, it's kind of pointless. Okay, so how do we do that? There have been some new tools which have been developed, which have been trying to address this problem. In part, they take advantage of the fact that the mice that we use in the laboratory are in fact related to each other. And they're related to each other because they were derived from mice. Remember, I told you about the mouse fanciers, the people who were using mice as pets. The way that these mice were generated is to make crosses. It turns out that the parental lines that were contributing to these were relatively constrained, and so in contrast, similar to the human bottleneck, there were bottlenecks in the generation of inbred mice. And then what that led to is basically haplotypes that are segregating in inbred laboratory strains. And they're different haplotypes in different mice, but the haplotypes represent permutations of the two main parental lines that went into the generation of inbred strains, mus musculus musculus and mus musculus domesticus. And you can see here a comparison between two different strains. And what you see is that where their haplotype is concordant, because they inherited the same parental alleles, you find relatively rare SNPs. And where they're discordant, you find a lot of SNPs. And this is actually on the same back, the same physical entity. So this rules out any artifact. These are truly recombination breakpoints where you have haplotype similarity and haplotype dissimilarity. And so this is similar to the kinds of linkage disequilibrium blocks that we use in human genetic analysis, and it allows us to fractionate the mouse genome with the aim of increasing the resolution and understanding the genes that are contributing to phenotypic differences. And so a recommended strategy to try to increase the resolution of QTL analysis was to do your genetic mapping. You try to refine it. And then what the idea is to do is to basically look at various crosses and find the regions of haplotype similarity and dissimilarity between them to fractionate the peak that you obtained into the regions in which the underlying causal gene must reside. That is that if you have regions that have haplotype similarity, that's unlikely to be where the gene is. It's not impossible because these strains have been separated and some spontaneous mutations occur, but just as a probability event, there's much more genetic variation in the regions of haplotype discordance, and so that's where you would look for your presumptive causal genes. And so this is analysis that we did in order to try to address the airway hyperresponsive phenotype using exactly this approach. This is a cross between a different strain. What I showed you before was the AJ strain crossed with Black 6. AJ is hyperresponsive. We established that the AKR strain is also hyperresponsive. We did the exact same thing. We got a number of peaks. We were very happy because these peaks on chromosome 2 also showed up in our previous analysis. These peaks did not. But the problem is that the resolution of these peaks remains very, very broad. And so even though we now have 2 strains, we have huge regions of the genome in which these causal loci can reside. They may be multiple loci under these peaks, and we're looking at the cumulative effect of them. And even using these haplotype strategies, we do not have sufficient resolution to even begin to contemplate what are the causal effects. So what do we do about that? Well, I personally, for one, have given up and have tried different approaches, but I'll show you some of the strategies in the field. This is that AKR cross Black 6, and on the important thing is on the left is the phenotype axis, and you can see the variation of phenotype in individual N2 mice. This is a cross between non-inbred mice, and so it's basically as a practical matter what people in the field have, or some people of the field, and said we're giving up. We're not going to use inbred mice. We're going to use the strategies that have worked so effectively in human disease. We're going to apply them to mice that are heterogeneous, outbred, and we're going to see if we can localize genes that way. And you can see some our foray into that looking at this airway phenotype. The heartening thing is that you actually see remarkable phenotypic heterogeneity in these mice. That's the good news. The bad news is that we didn't do any better at localization using genetic analysis, what effectively is an association analysis, because since they're outbred, we're not doing linkage in this. And okay, so let's see. So this is the outbred analysis. There is a group of investigators in the mouse that are interested in complex traits, and recognizing the limitations of these approaches, what they proposed to do was develop a resource for doing high-resolution genetic analysis, creating a new set of recombinant inbred strains. And just to let you know what recombinant inbred strains are, remember that we have inbred strains. What we can then do is create new inbred strains from these. And so here we're creating an F1. These are at heterozygous at every locus. And then if we take their progeny and we do brother-sister mating, what happens is they're going to fix loci from one parental strain or the other after sufficient generations. And so you're going to basically create mice that have mixed the deck from the parental strains, but then have now fixed that mixing, and so they've created, they've fragmented the haplotypes that are present in here into smaller haplotypes that are present in these recombinant inbreds. And so the group involved with this project, which is called the Collaborative Cross, took a very, very thoughtful approach to this, picked 8 parental strains that they wanted to create new recombinant inbred strains from, used a very systematic method of mating to try to generate progeny now in which all of these parental strains are being assorted, and sort of creating a pseudo-human population. Pseudo in regards to the fact that these are inbred mice, and so they're homozygous. And obviously the human population is not. But the relevance to this is that we now have small haplotype blocks. And the fact of the matter is if you want to create a real population that's similar to the human population, what you can do is you can take these inbred mice and you can cross them. And now what you have is what's called the diversity outbred mice, a very, very heterogeneous population genetically, one that is segregating all kinds of alleles. But what's important is that you can go back and you can recreate these. So you know what they came from, and so if you're interested in segregation, if you have hypotheses about what's doing what in an analysis of these progeny, you can go back and test that hypothesis. So this is a very powerful resource, and when you look at the phenotypic variation in this resource, it's huge. So that's the good news. There is a bit of bad news as far as I'm concerned, and that is what was done in order to maximize the diversity in this resource was including three what are called wild-derived strains. Most of the inbred laboratory strains that we use are the ones that are derived from the strains that I showed you earlier. But in order to maximize diversity, what people have recognized is that if you go out into a field and catch a wild mouse, you can inbreed that mouse with difficulty, but eventually, and you can create a mouse that is quite genetically different from an inbred laboratory strain. And that was what has been done, and these mice were included in this cross. And so that accounts for the kind of phenotypic variation that you see here. But the problem depends on how you look at this. A potential problem is that by introducing this genotypic variation, you may make the positional cloning task more difficult. And this is illustrated here just as a comparison in a random region of the mouse genome of SNP variation compared to the reference strain black 6. And you can see these are all the inbred laboratory strains, and you can see very clearly the haplotype blocks that I was talking about, where you get long regions of concordance and some regions of discordance between these different inbred strains. And here are the wild-derived strains that you can see. What you can see is they're completely discordant. So that you may be able to localize your trait to a small genetic interval using these mice, but if that phenotype is being conferred by wild-derived loci, which empirically appears to be frequently the case, then you still have a lot of genetic variation to sort through in order to assign causality to a specific locus. So this remains a challenge. Okay. The last thing I want to talk about is the reverse genetic analysis, and this is important because we can relatively readily modify the mouse genome and ask specific questions about gene function once we've perturbed the gene. This takes advantage of the fact that we can actually manipulate the mouse oocyte and do microinjection, and what you see here is an oocyte. It's being held by this suction pipette, and it's being injected using this glass pipette. You can inject DNA into that, or you can inject cells. And the original genomic modifications were done by injecting DNA. Simply harvest those oocytes. These are fertilized oocytes. Inject DNA into the pronucleus of the fertilized oocyte, reimplant the oocyte, and with some frequency that injected DNA is taken up, incorporated randomly into the parental genomic DNA, and you end up with a transgenic mouse. And so this was one of the first ones created in Phil Leder's lab, and you took the c-Myc oncogene, you took an enhancer that drives expression in the breast, put those together, inject them into the mouse, and created a mouse that develops breast tumors with a much higher frequency relative to non-transgenic mice. So there are certain advantages to this approach. It's fast. You can frequently generate a faithful tissue-specific expression. You can, as I said, sort of mix and match. You can drive genes and put them under control of different promoters. There are disadvantages, and one is that these things are being randomly put into the genome. They are put in as concatenamers, so you have different numbers of transgenic of genes in your transgenic. The expression isn't necessarily proportionate to the number of genes. So you have to look at multiple lines frequently in order to sort of establish some consensus about what the phenotype is of your transgenic. You can only test dominant effects because you're only adding DNA in this case, although you may have unintended consequences that are recessive that have to do with the fact that these genes or these transgenes can land in genes and they can have disruptive effects, what's called insertional mutagenesis. So there is a potentially much more powerful system in which the modification of the mouse genome can be done in a directed fashion. And that takes advantage of embryonic stem cells. We have all heard about embryonic stem cells. What you may not appreciate is all of our appreciation of embryonic stem cells derives from studies that were initially done in the mouse, and what they derived from were from studies of teratomas. And this is a teratoma transplanted in the mouse, and what you can see is that there's actually very well differentiated tissue. This is a tooth. There's all kinds of different epithelial and tubular structures in here, and people who were studying these recognized that these teratoma cells, which are embryonic tumors, were maintaining multipotentiality, not pluripotentiality, but multipotentiality. They could differentiate into all kinds of different tissues. And so this compelled a systematic investigation into these types of cells, the recognitions that if they could be multipotential, perhaps they could be pluripotential. And so at roughly the same time, these different investigators determined that they could go to the mouse embryo, a normal mouse embryo, not a tumor, and obtain pluripotential cells. And that's illustrated here. What they recognized is this is the developing embryo at the 8-odd cell stage. And then here you have a blastocyst, and within the blastocyst, you have what's called the inner cell mass. And what these investigators recognized is that if you basically obtain this inner cell mass and grow them, they retain not just multipotentiality, but pluripotentiality. And pluripotentiality means that you can differentiate into all cells, including germline cells. And that's the key point here. So this is illustrated how this is done technically. You take your blastocyst, you disrupt it, grow it on a feeder cell layer, obtain those inner cell masses I showed you, and then basically, you disrupt the blastocyst here, obtain the inner cell masses, grow them, and then these then become embryonic stem cell lines. And fortuitously, this works because these embryonic stem cells have very characteristic cellular morphologies, and so one could recognize these and manipulate them. All right, so how do we use these? Once you have an embryonic stem cell line, you can then reintroduce those stem cells into the developing blastocyst, and they are incorporated into the developing mouse. And that's illustrated here. Here's your blastocyst, your host blastocyst. Here's the embryonic stem cell line, which you have generated. You've generated it from a pigmented mouse. Your host, in this case, is from an albino mouse. Those cells are incorporated into the developing blastocyst, and you end up with a chimera. A chimera is a mouse that is derived from two different parents. This is genetically homogeneous within each parental-derived region. These are not F1s, these are not heterozygous. This is a mixture of two different strains of mice in one. And so then what do you do with those? You basically go through a cloning step, you breed them. And so you have this mouse, it's got a germline, if everything worked well, that is a chimera. It provides to each offspring a single cell from the germline, and if it came from the ES cell-derived region, now you have a mouse which is homogeneously derived at least one allele from this embryonic stem cell line. And this simply illustrates that microinjection step. And this is the chimera. You can see this is a chimera between a pigmented mouse and an albino mouse, and you can actually see where it's chimeric right down the midline here. And then hopefully its germline is chimeric, and you end up with 50-50 progeny of whatever it is you want to do. Okay, how do you use this for genome modification? I'm going to have to go fast here. Fortuitously, embryonic stem cells turn out to be relatively permissive for homologous recombination. And so what you can then take is a DNA sequence that has homology to a region of the mouse genome that you're interested in, and you can modify it, and then you can add that to embryonic stem cells. If you have a selectable marker, you can identify those cells which have incorporated the DNA and have generated a specific modification event. And once you've done that, then you can inject that modified ES cell line into blastocysts, get your chimera, and end up with a progeny that carries your genetic modification. This illustrates that event where you're disrupting a gene by basically creating recombination sites in the genome, so that when you end up getting your genome, it by itself is not modified, but it carries recombination events that, when combined with a transposase, will result in a modification. And there are various recombinases. Cre is the one that's most commonly used. If you take that modified allele that I showed you, cross it with something that expresses the recombinase, that then results in a recombination event only where the recombinase is expressed. You end up with that recombination event that results in a gene modification, in this case a deletion. And now what you have is you've controlled your gene modification. So instead of just having a knockout across the genome, you have a conditional knockout that you can control, and so you can ask much more specific questions. This is an illustration of a knockout of a gene, the sonic hedgehog gene. If you were interested in the consequences of a sonic hedgehog knockout in the brain, this would not be a mouse in which you could study that. But if you create your conditional allele, you can control that recombination event, and you can ask the question specifically in the brain, what's the consequences of recombination? Fortunately, if you're interested in this, there's been a international effort to generate these things. The International Knockout Mouse Consortium has embryonic stem cells in which these have been generated. You can identify them just by going to your mouse genome browser, pick your gene, and see the state of these things, and then you can just order them. So very quickly through that last step, but you can modify the mice. You can do reverse genetics in a very powerful way. There's some very powerful new tools using zinc fingers and talons to do this and ask very specific questions about point mutations. And so you have a nice complement to the type of forward genetic approaches to do this. So that's the mouse genetic primer. I think we're reaching the time for our MPG session. So perhaps we can save questions, if any. You can just ask me directly. Okay?